Okay, I have the screen now.
Green, and let me just kind of mention to the audience, if you have any questions, feel free to type them out in the chat pod. Dr. Tolleson will be covering the questions.
Okay. Well, first of all, thank you for inviting me. This is a nice opportunity, and I always look forward to talking about this topic. This is something I've spent quite a bit of time on. And what I want to do today is cover kind of a broad spectrum of topics dealing with nutritional monitoring of range animals. So we're going to kind of start with plants and that type of monitoring and move through the animals themselves. Talk about some old school methods as well as some new high-tech type of methodology that is either already available or should be coming online shortly. And I'm going to try to put all that together by the time we get to the end of the hour. I've got a lot of slides here. Some of them I'll kind of flash through because I think they're concepts that folks will understand, but I just want to kind of get us on the same page and be thinking similarly. But feel free to pop a question up there and stop me, and we'll take the time that we need. I want to start off by saying that these nutritional monitoring concepts that we'll cover are not going to just be applicable to Texas livestock. These are pretty much going to be applicable around the world, and we can apply these same concepts whether we're talking about domestic livestock like the Hereford cow in central Arizona or the domestic yak in northern Mongolia in the top two panels of this slide. Apply equally well to wild herbivores like the bison in southern Oklahoma and the impala there in Kenya. So some of you may be more livestock oriented, some of you may be more wildlife oriented, some of you may have interest in both, and so hopefully you're going to find some material that's going to be useful for you regardless of what your range animal type interest may be. A lot of what I'm going to talk about today, basically everything, is can be related back to this one slide here. Probably a lot of you have seen this or something like this, and you could pretty much teach a graduate range class off of everything that's going on in a pretty simple graph like this. So if we look at the quantity of forage that is produced by a given landscape and the performance of animals on that landscape, whether it be on a by animal basis, as we have represented here in the red line, or on a per unit area, so an acre, hectare, square mile section represented in the turquoise line, we can relate some indication of livestock or wildlife production on the y-axis to some level of increasing grazing intensity or plant tissue removal on the x-axis. So if we have a unit of land that's going to produce some adequate amount of forage for an animal to survive, as we start putting animals on that land, the animals themselves early on are going to produce basically at their genetic capacity to produce and what that land is able to provide for them. As we move to the right across the screen, as that red line starts to go down, as we add more animals and increase the tissue removal, then there's going to start to be some competition and maybe not quite as much forage available for each individual animal, and they may not produce at their genetic capacity. But we'll see that as we add animals, the production per unit area in the turquoise blue line is going to increase. Kind of that magic sweet spot that we're looking for is where those lines cross, and somewhere in that general area is where the art and science of range management come together to try to reach an area where the land is providing adequate nutrition for the animals. We've got the number of animals matched to go with the amount of forage that land can produce. And so the land per unit area is also being productive, and we're not doing anything to the detriment of the land or the animals. So a lot of what we're going to talk about today is going to be really wrapped up in this graph. So we're going to start off talking about the forage itself and monitoring that, as that's going to relate to our animals. And the first thing that we can do is measure the quantity. Forage quantity is going to basically be pretty simple. It's going to be a weight per unit area. We typically measure that in dry matter. We need to be representative if we're going to go out and sample pasture or landscape. We need to incorporate the difference that we might have in the type of range sites, the vegetation types, topography, distance from water, that sort of thing. And we need to realize anytime we go out and collect a measurement like this, that that's going to represent that point in time where we are in the phenology of the plants out there, the time of year, the season, where we are in the growing season from the beginning to the end when we get to a frost. Going to be pretty simple to measure. We're going to put some size of a frame or a hoop down on the ground. We're going to clip the forage down to close to the ground level within that frame. We'll weigh that, dry it, get a dry matter per unit of this small frame size, and then we'll do the appropriate math to calculate up to the pounds per acre or kilograms per hectare. So we're looking at about 400 pounds per acre with range that had about this much forage on it. And we can go up to some areas in East Texas, high rainfall areas, where we may get 3,000 or 4,000 pounds per acre. Now we can also measure and monitor forage quantity with some very high-tech methodologies that are going to be built off of old school. We're going to have to go out and clip forage and identify plants and determine what soils they're on and how much rain has fallen on a given site. But we can put all that together into systems like this global livestock early warning system, which is a Texas A&M product. This happens to be an Arizona map of the Coconino and the Prescott National Forest in central Arizona. And I'm going to show you a lot of Arizona data today. I'm been back in Texas at the Sonora Research Station for about 2 years now, but I was in Arizona with the University of Arizona out there for 9 years previous to that. So a lot of the data that I have, hands-on personal data, to be able to illustrate these concepts to you are going to be from Arizona. So, but I think again they'll still be applicable. In this particular instance, we're looking at a landscape about a 75-80 mile radius there from the center, and all the little colored markers that you see there represent a site on the ground where forage data has been collected, the plants have been modeled, and they are predicted on a day-to-day basis on the type of forage that's there, the rainfall that has fallen, and the amount of herbivory that we would expect, and kind of do that balance to see whether we would be above or below some long-term average. And so it's typical color ramp here. The yellow sites that you see there would be expected to be slightly below. The light green to dark green would be sites that would be above to greatly above. And again, this is going to depend a lot on the site itself, the elevation, how it's being grazed, and that sort of thing. These type of tools, they are publicly available, but not widely publicly available at this point in time. But this is the type of technology that is coming down the pike to be able to, again, monitor forage quantity. So for those of you that are in South Texas or in any other brush infested area, it's pretty easy to go out and lay a hoop down on the ground and determine how much grass you have, but determining how much forage you may or may not have in a brushy environment like this gets a lot more difficult. So we do also have some technology that we're working on, again, combining some old school and some high-tech methods of trying to be able to quantify the biomass that may exist in various types of shrubby species that, again, may or may not be providing forage, and we kind of need to know both sides of that equation. On the left side of the slide there, we can take a shrubby species and just go do some old school physical measurements. We can look at basal diameter, height, width, apply some calculations. We can cut and weigh and measure those trees, and we can come up with some pretty high correlation type calibrations to make a couple of physical measurements and then estimate the amount of biomass, total biomass of a plant and actual grazable or browsable biomass. We also have some more high-tech kind of space-age technology type methods represented here on the right side of the slide. The top image is a LiDAR kind of a true color representation of that tree that we see that we did the physical measurements on. And then, so a 3D point cloud representation that was determined by a LiDAR scanner. And in the bottom graph, the colorized graph there, is where we've applied some calculations to determine what part of that plant is actually available as forage. So if we figure a browse line is going to be somewhere around 5-6 feet, you can see that on the green, the lower portion of that tree, pretty much considered to be all available to most browsing livestock. The yellow area starts to get to where some of it is unavailable due to the height and maybe some of the density as you get into the plant itself, and then the red would be basically unavailable. So taking measurements like that gives us the ability then to go and do similar stocking rate type calculations for browsing animals that we would do, that most of you have had in a Range 101 type of a class or a workshop, where you determine how much forage is available on the ground and extrapolate that up to pounds per acre, put the number of animals you have, you know how much forage they're going to consume, and then you basically just do the math. How much forage do I have for what length of time, and how many animals can I run? So we're looking at trying to be able to do similar type calculations for browse. So forage quality is going to be the other half of that equation. We need to know how much forage is out there. It stands to reason then that we need to know what the quality, what the nutrient density, how much protein, energy, vitamins, and minerals those plants are going to be able to provide. I'm going to primarily talk today on protein and energy. Those are the macronutrients. It's not that vitamins and minerals aren't important. They certainly are. But that would be a whole nother 2 or 3 hour seminar just talking about vitamins and minerals. So we're going to concentrate on protein and energy today. So forage quality is going to be dictated a lot by the vegetative characteristics, the size, shape, point in the growing season that particular plant is at the time that it's being grazed. You'll see that animals, if they're going to choose forage, they're basically going to try to choose new growth over old. So it stands to reason then live over dead, and then leaf over stem. So if an animal gets to choose, they're going to choose new live leaf to get the highest quality of forage. They're going to consume some of the old dead stem as well to get some bulk and some fill, which they also need. Now the quality in those new live leaves versus old dead stems is going to be related to all the way down to the basic building block here of a plant cell. So the structure of that cell is going to aggregate up into the structure and the morphology of that plant, which is going to dictate the quantity and quality of forage. So if you think back to your kind of 10th grade biology class, you probably had to diagram a plant cell, and you would have had to put in the cell wall and a lot of these different organelles, the chloroplast, the mitochondria, vacuoles, and those sorts of things. If you think about the cell of a plant as providing a lunch to the microbes in the rumen of a ruminant animal or the hindgut of a simple stomached animal, and think about the different types of lunch or a meal that these plant cells may provide when they enter that rumen environment. In the new live leaf, in these young lush parts of the plant, they're going to have cells which have very thin cell walls and less chloroplast, or I'm sorry, less cellulose and structural carbohydrates relative to the organelles and the cytoplasm that are inside the plant cell. So you're going to have less fiber relative to the protein, the enzymes that are providing that nutrition to the microbes. If you think about an older plant cell on a stem or some of the dead material, you're going to have the reverse. You're going to have thicker cell walls, more cellulose relative to the nitrogen, the protein in the cell. So that's going to be more like a box lunch that you're providing to those animals or to the microbes. It's going to be harder to break down, take a little more energy, take a little more time. So young lush forage is going to be more like a sack lunch for your microbes. Old dead stem is going to be more like a box lunch. So when we look at a landscape level then, or a pasture level, in a range situation, forage quality is going to be a mosaic of different offerings for that buffet to those animals as they go out and start to graze. If we see some of the, this is a graph from the Noble Foundation on some warm and cool season range plants. If we looked at something like a Plains blue stem here, we can see that the growth curve of that plant is going to grow in May, June, July, be a little peak again in September if we're getting rain like we've been getting recently. And you can look and see similar growth curves for those other warm season type plants. When we move down to the cool season, again, we'll see some areas here where during the early part of the year, the cooler times in the spring, those cool season plants are going to put on their growth. They're going to go a little more dormant in the summer and then pick up another growth curve and a peak in the fall. And so when we talk about a range situation, and we have our general category here of native grass, we're going to have a variety of plants that are coming and going at different stages of phenology at different times of the year. And hopefully then that's going to provide a nutrient environment for our animals that's going to be providing some quality at different times. So I'm going to show you an Arizona graph again. And I think this does some good illustration for us here. The shrubs are going to be in the red line and grasses are going to be in the black. And if you'll remember that Arizona and the American Southwest has a monsoon weather pattern, so typically dry during the late spring, early summer, and then we get the monsoon that comes in July, August, and September, and that's where we get the peak of our growth. So you can see that in the protein level of plant tissue that was harvested and chemically analyzed in Arizona. We'll see this, low quality January, February. We'll get a peak of quality in April and May, but realize there won't be a lot of quantity at this time. It'll be young, lush, growing material, but we won't have the amount of rain to produce mass in the early part of the year until after this early summer drought, and then we pick up into the monsoon area. And that's when we're going to get the both quality and quantity occurring at the same time. So I want to show you some different species of plants and the protein that they would provide relative to the requirement of a moderate size, moderate milking beef cow, and look at some different levels of tissue removal. And so to kind of make the point that the way we manage these plants is also going to have an effect on their ability to provide nutrition for the animals. So if we take four-winged saltbush, which is a shrubby, small shrub type of a browse species up in the Arizona Strip, which is north of the Grand Canyon, between there and the state of Utah. We see in the dotted line here, we see the protein requirement as it might change for that, again, moderate size, moderate milking beef cow that would calve probably in March and be weaned in October. And we can see that even at a heavy level of grazing here or browsing in the red line, this four-winged saltbush is going to provide protein above the requirements for that cow throughout the year. And when we get into a moderate or a lighter amount of tissue removal, the tissue that's left in that plant is still going to be providing above the requirements for this cow. The caveat here, though, is that the amount of four-winged saltbush on the range is going to be distributed in such a way that it's not going to be providing the bulk of their diet. If we look at a cool season grass, Indian rice grass here, looking at the same situation in this graph, we can see that in a more heavy tissue removal situation, that red line, very seldom is that grass going to provide a quality of protein above the requirement for that animal. But with moderate and light grazing, most of the year that grass will meet their requirements. And then if we look at a warm season grass, and again looking at this protein requirement for the cow, we can see that with heavy utilization, the galleta, similar to tobosa grass, not very often is it going to meet those animals' protein requirements, and it's going to take a more moderate or light grazing um, for in that very dry, harsh environment up there in the Arizona Strip for that forage to provide the quality of protein that those animals would need. So let's shift gears a little bit now and let's start talking about the animal side of this and how they're interacting with the forage environment. And different species of animals are going to have different foraging strategies of how they go out and acquire forage, and then they're going to have different digestive strategies of how their gastrointestinal tract is going to handle that and turn that plant material into nutrition for them. And so if we're talking about a horse in Mongolia or a white-tailed buck in central Texas or a giraffe in Kenya, they're going to be very different at the way that they acquire and then process forage. This is going to relate back to some pretty simple anatomy. The mouth parts, so form and function, are going to be related here. If you've got mouth parts that are, you have a muzzle that's very narrow with split lips, long tongues, like we might find in a goat or a deer, they're going to be able to be more selective of very small plant parts. Typically, these animals will have salivary glands that are going to help start that process of digestion of some of those maybe highly nutritious but perhaps have some anti-quality compounds in them, like we find in some of our brush. We get into the animal, the relative proportions of the reticulum and the rumen, or the cecum and the hindgut, whether we're talking about ruminants or simple stomached animals, hindgut fermenters. And then there are going to be some other anatomical considerations, and we're looking at just the gross physiology of that animal, whether they're going to be in a quadrupedal, four-legged grazing stance, or they have the ability to have a two-legged or a bipedal grazing stance. So the overall morphology of that animal. As we look at this bison with a very large head, wide mouth and muzzle, large jaws, that big hump there to anchor that head. We can see the large rumen and the paunch area of that animal. They're able and built for harvesting a lot of forage material, holding on to it for a period of time and extracting all the nutrition out of it that they can. And so they're kind of a mowing machine as they're moving across in a four-legged stance. We can compare that to a white-tailed deer, which is a much more of a tubular shaped animal, has that ability to raise up on two legs, harvest a different horizon of forage as opposed to those that are more grazers. And so this can be a competitive advantage then, to them to be able to access forage that maybe some others in a particular grazing guild may or may not be able to get at. So we can break our livestock and our wildlife down then into some different forage strategy categories based on how much grass versus browse that they consume. So if we look at some of our primary livestock species here, cattle, sheep, and goats, cattle are going to be more on the grass grazing end of that spectrum. Sheep are going to be more intermediate. Goats are going to be more on the browsing side. We can look at wildlife and break them into some of the same characteristics or categories. Bison are going to be more grass browsing type. Elk are going to be much more intermediate and very plastic. And moose are going to be more on the browsing side. And this brings us to a concept where we'll talk about diet plasticity or overlap. And when we've got multi-species livestock grazing, when we've got livestock on the same range as wildlife, when we have multiple types of wildlife, and especially some of the exotics that we have, these are going to be some concepts that are going to be pretty important as we're trying to plan for the amount and the quality of forage that's available out there, and adjust our stocking rates looking at our carrying capacity. Diet plasticity just means that an animal has the ability to move back and forth between these different broad forage categories, from grass to forage to browse, things like sheep, things like elk, species like axis deer. So they're very plastic, and they have the ability to utilize a wide range and a variety of forage, and this is one of their competitive advantages, where something like a bison would be more on that grass end and be a little more obligate toward the grass. A moose or something like that would be more on the browse end. And so overlap then would be the amount of competition for forage that different species may exhibit. So let's look at some graphs here, looking at some of this diet plasticity and diet overlap concept. If we look at an overall for a year diet composition by forage type from our cattle, sheep, and goats, and not surprisingly, we would see that the bulk of the cattle diet is made up of grass, some forbs and a little browse. Sheep, a little more of an even distribution, and goats, again, probably even more even between the browse and the grass, but much more browse than either of the other two species. If we look at it on a similar three wildlife species, we can see again those bison are pretty much obligate grazers, do consume some forbs, and in this case they documented some browse in the dark green bar there. Elk showing a little more plasticity, similar to what we saw with the sheep, and then the mule deer being pretty much the opposite of the bison in this case, pretty much obligate browsers, but with some ability to use forbs and grass. Now these are general annual consumption represented here in these graphs. We can break this down by different periods, and we'll look at season in this case for one species, and we'll look at cattle. Same type of bars, the grass in the bright green, the forb in the olive, and the browse in the dark green. We can see that in the spring, the cattle were, in spring, summer, fall, and winter, were predominantly grass. A little bit of browse during all of those seasons, and the forb component varied depending on what season we were in. And then we'll see a similar type of arrangement for sheep. Pretty consistent with the amount of grass across the seasons. The forb and the browse component changed. Not rocket science here. One of the reasons that they ate more forbs, both the cattle and the sheep ate more forbs in the spring, is because that's when they were available. One of the things that I've noticed about both livestock and wildlife throughout the past 30 years or so is that they don't come to very many of our workshops, and they apparently don't read our books, and so oftentimes they will eat things that we don't expect them to eat, and they will basically eat what is available to them, and they will also experiment with different forage types. So this particular mule deer doe here, picture was taken, I followed her around for about 30 minutes, about 8,000 feet in eastern Arizona, and she did nothing but eat mushrooms. So that's not a bagel in her mouth or a donut. It was a very good mushroom year that summer, and mushrooms have a lot of protein, have a lot of different vitamins and minerals, and this was apparently something that this doe was specifically seeking out because I saw her consume quite a few of those. And similar, this is a Hereford beef cow in the fall of the year at about 5,000 feet in central Arizona. She's got winter grass, side oats, several drop seeds, blue grama, and plenty of forage available to her, and she spent about 30 minutes grazing turbinella oak, and not particularly attractive looking turbinella oak. So there was something that she was looking for in that forage, and when she had other quote-unquote better forage available to her, she was still going after this. So the point here is that we have a lot of data about what animals eat and why they eat it, but sometimes they won't necessarily go along with our trends. So let's shift gears again now, and let's start talking about how animals will use a landscape, how they will acquire forage, with the idea that if we're going to monitor this and we're going to plan where we're going to go and collect forage and to do our stocking rate calculations or to analyze this forage chemically, we need to understand how the animals are using the landscape. So it's going to go from the bottom of the graph actually and work up. Animals are going to select plants and species and parts within those plants. If they stay in one place with their feet in place, that selecting plants, that's a feeding station. As they start to move from station to station, they'll utilize a patch. Patches will aggregate up to communities, and communities to whole landscapes. And you can see that here. If we start down on the lower right portion of the graph, again, an animal would start by selecting and interacting with the forage environment at that individual plant and plant part level, aggregate plants together to a feeding station, to a patch, to a plant community, and then again up to the landscape. So they're going to select these plants then based on some different plant preference categories. And I want to apologize that when this slide came through, there's some text that's missing here. So down in this area between the graph and the words of performance, maintenance, subsistence, and toxic, here we would have performance is going to be provided by plants that are preferred. And so the green bar here is the percentage of plants, and the axis, the percentage axis, also didn't come through. But preferred plants are going to be found in the diet in a higher proportion than they are found on the landscape. Sometimes we refer to these as ice cream plants. It might be those mushrooms that the deer was going after, something that is providing a very high quality nutrient item for them that they need, and that helps enhance their performance. Over in the maintenance category, these are plants that are going to provide a lot of the bulk of the diet. They're going to be consumed, as you can see, those two bars, the green bar representing diet and the turquoise bar representing what's available on the landscape, fairly equal. So they're going to be found proportional in the diet to what's available on the landscape, and this is going to provide a lot of the maintenance for the animals. If we move over into less desired, sometimes forced categories, plants that when we run out of the other two categories, we're going to start eating some of these. Threons might be a plant that would fit into that category. They will provide nutrition, they're just not highly desired. And then we can move over to toxic plants that can be injurious to the animal, and typically they're going to be consumed in less proportion of the diet compared to what they're found on the landscape. So let's start talking about the animals themselves now. So we've covered some ground, literally and figuratively, talking about plants, talking about the quantity of forage that's available on a given landscape, the quality that those particular plants are providing, the way that the animals will use a landscape to acquire those plants to try to meet some of their nutritional needs. So let's talk about some of those nutritional needs in the animals. This particular animal was one of our Hereford cows on the V-Bar-V ranch in Arizona, and she was voted by her classmates as the most likely to become a rug at some point in time. A very pretty little cow, raised a good calf every year. She's going to have every day, just to meet her nutritional requirements, we're going to start off with maintenance. Her heart's got to pump. Her lungs have got to bring in and expel air. Her intestines have to contract and move food along her digestive tract. She's got to have muscle movement. The bones have got to have the ability to hold her up and move her around the landscape. So we have to meet those maintenance requirements every day. If we're going to be productive as livestock people, we've got to have these animals reproduce. If it's a wildlife, they've got to reproduce and continue their species. If we're talking about mammals, these animals are going to have to lactate, produce milk. This is going to be the biggest nutritional drain that they're going to have. If we've got a young animal, we need them to grow either to maturity so that they can reproduce, or to some market threshold that we're looking at, some weight and size and amount of fat to produce a good marketable meat product. In other parts of the world, these animals are also a lot of times going to have to do work. You may have to pull a cart and take supplies back and forth to town, as well as growing and lactating and reproducing. And then if we're talking about Angora goats or wool sheep, yaks, these sorts of things, and on top of all of that, they're also going to have to produce fiber. And so all of these require some amount of nutrition. And I just wanted to throw this slide in to say that, again, the same thing is going to be true for wildlife species. And so if we're going to look at a reproductive rate in something like an elk, if we look at the amount of fat or the amount of gross energy that's provided to them from their environment, the better their nutrition and those needs are being met, the more successful those animals are going to be at reproducing. So this slide is just to kind of bring us back to that first slide about the different species of animals in different parts of the world, and just remind us that our concepts are going to work here for wildlife as well as livestock. So if we're, again, talking about young growing mule deer, talking about some feral horses, that we may be as much interested in what effect they're having on the environment as what the environment is providing for them nutritionally. If we're talking about species that have very distinct reproductive seasons, like a rut for a bull elk, their nutritional requirements are going to change during very specific times of the year. And then we're going to look at animals like these pronghorn up at about 5,000 feet in the middle of winter. We've added a thermal aspect to their nutritional requirements. They've got to stay warm as well as acquire food to meet those nutritional, maintenance, reproductive type needs, and they're going to have to do it in a limited environment now, because it's going to have to be what they can find that's above the level of snow. So there are a lot of different complications that can, we have to deal with when we're talking about matching forage environment to an animal's nutritional requirements. And that's really what we're trying to do. We're going back to finding that sweet spot between those two lines that crossed in that production per animal and production per area. And to get this to be at an economically sustainable or an ecologically sustainable manner, we're going to have to match the requirements of those animals with what the land is able to produce for them. And so basically we can boil that down into a conceptually pretty simple, easy to say but harder to do graph, where we see that nutrition for the animal is going to turn into body condition, some indication of how much nutrition they're getting, and then that's going to turn into performance. Probably every extension agency of all 50 states probably have a graph that they've produced that looks something like this, and you can find them from various research organizations around the world, and they're all going to look basically the same. The height of the bars may change a little bit, and they may use a different body condition scoring system. But basically, if we're looking at the percent of animals that would be pregnant in the fall, let's say, of palpating with cows, compared to what their body condition was at the time they calved, the lower condition that they're in on a numerical scale, with 1 being emaciated and 9 being overly fat, 5 being a good average, the lower body condition animals are still going to reproduce, but at a much lower rate than animals that are up in that moderate to good body condition. And as we carried this graph over to an 8 or a 9 body condition, we'd see a flattening out, if not a dropping off of reproductive performance, as the animals can actually become over-conditioned. So this is kind of what we're looking for. If we're talking about beef cows, this is kind of a mature beef cow here. You can see some big calves out there in that herd. She's in pretty good working condition, fairly smooth. Got a little bit of a bag on her, got some grass out there, not a lot of green grass, so there's probably more box lunches than sack lunches out there for her rumen microbes. But she's been through the year when she was milking and producing that calf, and should have had pretty good nutritional environment available to her. If we're going to look at body condition as one of our techniques that we can use to monitor nutrition and see how well we're doing out there, which is probably the most common form of nutritional monitoring, it's going to be based off of anatomy, and we're going to look at some landmarks. And I like to use a pretty simple system. I spent a lot of time in Arizona teaching federal land management agency people who didn't come from a livestock background, were more botany, soil type education, teaching them how to body condition score. So I tried to make it as simple as possible. They're intelligent people. They just didn't come from a livestock background. So a lot of times we would look at the ribs first, and we would come back and look at the fat deposit around in the pelvic area. We would look at things like the short ribs, the transverse processes, and then we would look at the tail head when looking from the back. Now there's other areas here, brisket neck, things like that we can look at. But again, I felt like this was a pretty straightforward and simple way of teaching body condition scoring to people who, again, didn't come from a livestock background. So these are pictures that I've taken from an Arizona Extension publication. And so, again, we're looking to look at body condition and using it as a monitoring technique. We're going to start looking at these ribs first. And that's simply going to look at that and call it the rib effect. Number 2 is an area we're going to call the UV effect. And we're going to basically look at the shape of what you might see in a curve here when sunlight is striking these animals. And so in a fleshier, higher body condition animal, more fat deposited there. As the sun is striking that animal, you're going to have a shallow U, or maybe even no U at all, possibly flat in a higher condition animal. In a less body condition animal, you're going to see a deeper U or even a V. And that's going to be lower body condition. So we call that the UV effect. Next, we're going to look at what we call the TP effect. And that's looking at them from the back over the tail head. And if you're seeing a very sharp angle here, like the shape of a TP, that's going to be lower body condition. If it's more flattened out and you can't see that TP, then that's, simply that's going to be an animal that's in higher body condition. So this is an example of what we were referring to here as a 4. And I know on these next 3 pictures, there are going to be some of you, I recognize some names out there, and you're probably going to argue with me up or down a little bit on some of these. But again, our purpose there was to get non-livestock oriented people to be able to see big differences. And I think the 4, the 5, and the 6 are where most of the biology, most of the ecology, and most of the economics happen. And so if I could get them to recognize between a 4, a 5, and a 6, that was pretty much my goal. So we'd call this animal a 4. This animal we're calling a 5, and she's probably, in my opinion, a little on the higher end of the 5s, but we still can see, a little bit of rib here, a little bit of definition on the transverse processes, but she's pretty flat back through here, very shallow U. And then this would be the fleshiest cow of the 3, not seeing any rib, fairly round, fairly flat, pretty flat through here. Wouldn't be seeing any of a angular TP type of effect in this cow. So just monitoring body condition score is going to be one of the most effective ways of keeping up with nutritional condition of our animals. It's not uncommon for body condition to go up and down during the year, especially in range environments, and especially as you move further west and south into a little more arid, semi-arid type of country. So this is an observed body condition score for 1 year of animals in central Arizona, and you can see that they varied by about a body condition score through the year. These cows would have calved, here in this February-March time frame, calved in reasonably good condition as they begin to milk. And again, this is during that high quality but not as much quantity time of year as what we would have seen in Arizona. Getting into peak lactation during that summer drought, and then as lactation curve began to drop off and the monsoon began to kick in, then we started to see the body condition improve, and up until they were weaned, and then their nutritional requirements dropped off quite a bit, but the winter environment was a little harsh at this particular time, and so they did drop off in some body condition in these cows. So body condition scoring is again, a great tool, a good way to monitor animals. It gives us a lot of information about what's going on out in a pasture. The way that I learned how to body condition score and the way I started doing it, I'd carry a little pocket notebook with me and I'd go into a pasture and I would just start writing numbers down. 4, 5, 4, 6, 5, 4, 5, 5. Add up the number of animals that I had scored, add those scores up together and divide by that number, and I got an average body condition score. Dr. Stuth, who's passed away now, but a gentleman I worked for several years ago, taught me how to do this in this way. He said, put your categories that you expect to see out there, the scores, and then put tick marks every time you see an animal that's in that body condition score. And by doing that, then you create a bar chart, and you can see the distribution of the scores in the herd, and things like this will jump out at you that, yes, if we look at that herd on the average, they're about a body condition 5, a little better, pretty good shape for where they are at this time of year. But this will tell us that about 20% of our animals are actually in that suboptimum level of body condition. Now, if they're in a 10,000 acre pasture and we're only going to gather them when we brand and when we ship, there may not be a whole lot managerially we could do about that, but we still have that knowledge that when we get to the fall and it's shipping, this can inform us about what we might be expecting when we go in there and palpate. And should we have another gooseneck ready to have some cows, well, maybe we're going to have a few more that we're going to cull. If we're in a situation where we're gathering and moving animals a little more frequently, we have some alternate pastures and some things like that, maybe we can peel those animals off and manage them a little differently. But by collecting the data in this way, we at least know that. So again, you can do this pretty simply. You can do it in a spreadsheet or do it on your tablet. I know there are a lot of apps now that are available to keep up with body condition score, but you can do it pretty old school if you still want to do it that way. Now we're going to get to the really fun part of nutritional monitoring and some of my favorite topics. And so I always call this, you can learn a lot from a, insert your favorite word there, but we can really look at fecal material, cowpies in this instance, look at the forage that's coming from, and we can get a good idea about the nutritional environment for these cows and what's going on inside of them. As these cowpies stack up, the nutritional environment is going to be lower as they hit the prairie and spread out. And if you've ever been behind a cow in a wheat pasture, you know what I'm talking about. The higher quality, the diet that those animals are on, the more that's going to hit the splatter. So we can really learn a lot, and a lot of old cattlemen will do this. They pay a lot of attention to the condition of the manure in their pastures. And the reason for this is that the chemistry of the food that goes in the animal is going to be related to the chemistry and the physical properties of the feces that come out the other end. Now, especially in a ruminant, there's going to be a lot of processes, both metabolic and chemical, as well as just physical processes of chewing and the rumination and breaking those down into smaller particles, the microbial action. So that material is going to be quite different from one end of the animal to the other, but they're still going to be chemically and physically related. Fecal material is pretty complex, made up of a lot of different components. We tend to think of that undigested or indigestible feed stuff as being a major component, and it is. But in a ruminant animal, bacteria, both live and the dead carcasses of bacteria, can be anywhere from 30 to 60 or 70% of the dry weight. And so that bacterial environment that came from the rumen, that washed down through the tract, new bacteria that were acting on this digestive and the chyme as it flows through the intestines are going to be a big component of what's coming out the back end of those animals as well. So we can use that relationship of chemistry of the food related to the chemistry of the fecal samples by applying a technology called near-infrared spectroscopy, which we relate known diets in animals to their fecal chemistry. We can build calibrations such that once that calibration is built, we can go out and just collect the fecal sample, analyze that with the spectroscopy, and then predict what the diet of the animal that produced that feces would be. So in this graph, again from Arizona, over about a 12-year period, 15, 20 ranches a month sampled during this time period. Crude protein is depicted in the solid line, digestible organic matter in the dotted line. And you can see a very similar curve here to what I showed you from the plant material that was collected in Arizona. Lower during the winter months, a spike in quality during the spring, early summer, that early summer drought, and then the monsoon kicks in, and then we get quality and quantity through the late summer into the fall, and then we see it flatten back off as we go into the fall and the winter. So we've got two different measurements here. The earlier slide that we showed the grass and the brush and those two different peaks of protein on chemical analysis of plant material, and a very similar relationship here to what we saw with a spectroscopic analysis of fecal material. So again, the chemistry of what went in, very related to the chemistry of what came out. So let me show you a practical kind of a case application of using this technology and putting the fecal sample analysis in with a nutritional balance, a computer model that balances the nutritional needs of an animal with the forage environment that they're on, as indicated by the fecal NIR. We did this on the V Ranch, a University of Arizona experimental ranch in central Arizona. We went from about 3,500 feet of elevation down on the desert end of the ranch to about a mile high, about 5,000 feet in the central pinyon juniper portion of the ranch, up into the ponderosa pines at about 6,500 to 7,000 feet. Now this is a very unusual shaped ranch, but it's primarily because we had about 1,015-foot, 100-deep canyon on the south side and a similar canyon up on the north side. And so this allotment or this ranch went between those two canyons and basically used the different climates and the different vegetation zones going up and down the mountain. We grazed down here in the winter, here in the spring and summer, weaned up here at our headquarters in the fall, turned around, came back down, and we were back down here in the winter. So if we look at monthly fecal samples that were collected on this ranch for about a 2-year period, you're going to see that same pattern play out that we saw in the tissue samples, plant tissue samples that were collected, and in the overall Arizona fecal samples, and both of those look very similar. So we'll see that same pattern here on one ranch and one herd of cows collected monthly. So our early year winter samples, low in protein, solid line, digestibility in the dotted line, both picked up in the spring, dropped off in the early summer, picked up in the monsoon, dropped off in the winter again. Same pattern in the next year, from 2008 to 2009, spring, summer, late summer, but you'll see that the magnitude, the height of these bars is less in the 2009 year as compared to 2008. This year was wetter. This year, growing season, precipitation was lower. So we're seeing the quality of the forage as indicated to us by the animals, following very closely the expected weather and precipitation patterns, and as we moved up and down in elevation there on this ranch. Okay, we got 2 slides of the same thing. We'll move on. Now, when we take that information that we collected from the fecal samples that was telling us the protein and the digestibility of the diet that they selected, we put that into a nutritional model where we describe those nutritional needs of the animal. So we know how big they were, we know how old they were, we know roughly what their milk production would be, we know when they calved, so we know where they are in that lactation curve, we know when we weaned them, so we know when the lactation dropped off. We can compare what we collect and determine the forage quality to be and quantity, and put that into a model that determines those nutritional needs for the animal and then projects what their body condition would be given that set of conditions. We did this for two different groups of cows during a 2-year period. One was purebred Hereford cows. The observed line in the black here was going out when we collected those fecal samples monthly and visually collecting body condition scores for that herd. The red dotted line is what we would project then from the fecal sample and the nutritional balance model to what we thought the next 30 days body condition would be. And you can see that we kind of stayed within about a half to a quarter body condition and followed that trend throughout the year on the Hereford cows. We had a composite group of cows that came out of Miles City, Montana, Red Angus, Simmental, I think maybe some Gelbvieh in these cows, I don't recall exactly. Didn't do quite as well with them. There were some times here that we were about a body condition score off. Other times when we were, pretty much tracking them, and the general trend was about the same. But what I wanted to point out here is that if you're monitoring the nutrition of a set of grazing animals with a tool like the fecal NIR, or if you're collecting forage samples, there's going to be an iterative process here, and you're going to get a little better as you go along. But you're going to find times when you're going to see something like this, and you're going to be off. And so when you collect a sample that tells you something different than what your eyes are seeing, then you need to go back out and try to determine why that is, maybe collect more samples, get more data from these animals. Because if you will do that, when you come back, the next year or in subsequent years or subsequent seasons, you're going to get a lot better with any type of a monitoring process. In this case, we came back with these Hereford cows. We had basically they were phasing out those composite cows, and we didn't have enough of them to sample in the second year or in the validation part of this experiment. But with the Hereford cows, just during a growing season, so basically from breeding to weaning on that same ranch, working up that elevational scale, we got a lot better as we learned more about those cows and described them better in the model. So let's finish off here and talk a little bit about drought and how that's going to have some effects on our ability to monitor the nutrition of animals on rangelands. We know that as droughts progress and happen, we're going to move from green forage to dry dead forage. So we're going to go from sack lunches to box lunches pretty quick. And in the case of what we've seen at Sonora in the last 30 days, we're going to make a complete turnaround from basically dry dead forage to young lush, almost spring-like forage, because we've gotten 5 inches of rain there, a little over 5 inches of rain in September. So I want to point out a paper that was done back in 2010. A Kansas State scientist approached us at the fecal NIR lab there in College Station and took about 12 or 15 years of our data, over 20,000 samples that we had geo-referenced. We knew where they came from and what date they came from, the GPS point on the map, and did an analysis of warmer, drier climates versus moderate climates versus cooler, wetter climates. And not to anyone's surprise, we determined from this large data set that high temperature climates are going to result in generally lower diet quality, and low precipitation climates are going to also result in low diet quality. So if our climate projections are true, and we know we're going to have much more variation, but if we're moving into warmer, drier areas in much of this country, we're going to expect lower diet quality for our animals as time goes by. So we're moving into a time where I think nutritional monitoring is going to become more and more important, and I think we're going to need to be able to use all of the tools that are available to us, whether they be old school observational tools like this. We've got depicted in these slides here, looking at forage, looking at manure, being out on the ground. Boots on the ground are going to be very important. We're going to see situations where we've got dry, dead material. This is more of that box lunch situation. We're going to see fecal material that's going to stack more because there's more of that cellulose that's coming through, more fiber, as opposed to having young, lush green forage where we're going to get fecal pats that are going to be flatter, more spread out, and looser. And we're also going to need to be able to put this type of information together with some of our remote sensing, some of those tools like that livestock early warning system map that I showed you earlier. If we just look at a radar and a greenness product here, this is NDVI. So this is actually not the true color or the green color NDVI. This is a product that's going to give us a color ramp, which it's going to be a little difficult for you to see that on this slide, but it's going to be pretty typical of most color ramps you see, where white is going to be an average greenness, so amount and quality, photosynthetically active vegetation. Blue, and the darker the blue, is going to be above average for that point on the map at that time of year. Orange to brown is going to be the opposite. It's going to be less than average greenness for that point on the map at that time of year. And so I want to go back and think about that, whether it be the Arizona graph that had the plant tissue that showed the peak in the spring, peak in the fall, or those fecal NIR graphs that again showed how protein and digestibility were higher in the spring, lower in the early summer, and greater in the late summer. But I want us to think about all of this now in the context of not only where we are on the map, but what time of year we are relative to the phenology and the growth curve of those plants, the nutritional needs of those animals, and put all of it together. So from May of 2009, of that year where we had the Arizona data, we can see that most of our map here on the ranch is in white to blue. There are a few areas that would indicate being below normal, but our diet crude protein based on the fecal samples was right about 10,000 or 10%. Our cattle were at about 5,000 feet. Their body condition was a 4.3, and they were at about 60 days of lactation. So even though our map looked like we were in pretty good shape, we're above average from a remote sensing standpoint on the amount and the quality of forage that we would have at that point in time. Our cattle were physiologically in a stage where they had great nutritional needs. And so even though we had pretty good forage conditions, they were at that point dropping a little body condition because of where they were. They were raising that calf, and even though this is above average of where we would typically be in May, still below the nutritional needs of those animals. We can turn that around into September, where the crude protein of the diet was 12%, but we were at a time of year when we probably should have been at maybe 14 or 15% because we're late in that growing season and at an elevation where we were actually in a shorter growing season, so should have had pretty lush, pretty actively growing forage at that point, just about to get to the point where we're going to get into a frost and we're going to start to see that drop off. Cattle were at about 68,000 feet, 100 feet. Body condition is a little over 5, and they're approaching the end of their lactation. We're going to wean them in about a month. So even though our remote sensing, our knee-jerk reaction would be that blue is good and brown is bad, we have to, again, take it in the context of where we are on that point on the map, where we are in the growth curve of those plants, and where we are in the physiology and the production stage of those animals. But we can put all of that together. Again, look at our observation. What do the plants look like? What's the fecal material looking like? What's the remote sensing telling us? What's the body condition of our animals? And all of that can get us into a kind of a full nutritional monitoring mode using all of the tools that are available to us. So to summarize this, in the context of where we are at our range or our ecosystem site, the amount, the timing, distribution of precipitation is obviously going to affect our forage quality and quantity. Rain makes geniuses out of all of us. Animals are typically going to select that new live leaf, that sack lunch, as opposed to the box lunch, but they're also going to need to have fill. And so the climate, the weather is going to dictate how much of those higher and lower quality plants that are out there, the quantity of them. And then when we're thinking about our animal requirements, if we've got appropriate forage quantity and quality, that's going to provide adequate nutrition. Adequate nutrition is going to be manifest in body condition, one of the ways that we can monitor our nutrition. And that then is what's going to give us our economic and or ecologically sustainable level of production. So with that, I will stop and take any questions.
Doc, you have any questions, y'all feel free to type them out in the chat box. Well, we'll wait for the questions to come up. Let me say, I'm going to go ahead and kick out a survey.
Okay.
Please fill out the survey. I see somebody typing a question. Let me go ahead and kick it out. If it isn't pop out on your screen, I'm gonna go ahead and type it in chat pod as well.
Okay.
One thing I'm gonna mention as we wait for the questions to come in is that again next month we actually don't have a webinar. I'm gonna put some recorded webinars online. So if you need to pick up some CEUs, you can go to our website, and that's resourcewebinars.mu.edu, and you pick up some CEUs. And if you're not following us, you can follow us on facebook.com/txrange. I see you have a question there, Doc.
Okay, I'm seeing the survey, so I can't see the question.
Okay. The question is there a place to go get Excel documents to document all of our data easily, like you did on the form graphs?
Yes, there are several different apps that will have either a web form that you just fill out, or more of a spreadsheet type of data entry, and then I think either any of them you can probably export that out to an Excel or a text file. Gosh, I'm trying to think. There were several different body condition apps that are out, and I'm trying to remember. Probably some of our livestock specialists may chime in here. I'm drawing a blank on. I think maybe Cargill had one, maybe Hypro. But if you Google, cattle body condition, you're probably going to see some of those apps come up, and I've seen several different ones. Some of them that are part of just an overall cattle record keeping system, kind of an app version of our old red books that some of us may still use. So apologize for not being able to give you a specific name right now, because I'm having a senior moment and I'm forgetting. They're not coming to mind, but there are a number of those out there, I guess, bottom line for that.
If you look down your, he says thank you.
Okay.
If you look down your taskbar, you see a little green little looking icon. If you click on it, it should bring you back to.
That's got me back. Good deal. I will say on that the previous question too, that there are that I know of 2 grant proposals that are currently being submitted that will provide a dashboard to do, I think, exactly what Mr. Jones was asking for there. So depending on if those get funded or not, there will be a potentially kind of a whole range pasture animal management dashboard built and put together that would probably come online in the next 3 or 4 years. There was also a program put out, I want to say 7 or 8 years ago now, that was called FRAMS, Forage Risk Assessment and Monitoring System, I believe, that the range department at A&M put out, that was a monitoring record keeping system, I believe, like what maybe Mr. Jones is talking about, where you could put in body condition, you could use some of these monitoring tools, you could do some basic economic analysis. And so um, I'm not sure if that's actively available now, but you could certainly ask about it. So FRAMS, F-R-A-M-S, I believe you could Google that and probably get some information about it too. Other questions?
No other questions. I'm gonna go ahead and stop recording.